All right, so now that we've taken a look at what a virus is and how it can go through different cycles, its different life cycles, let's take a look at how we actually take a look at viruses and how we learn about viruses, culture them, and identify them. So first let's talk about isolation of viruses. So unlike bacteria, many of which can be grown on artificial nutrient medium, just like we can with everyday organisms grown on petri dishes in agar plates, viruses are not able to do that. So remember, viruses have to be within a cell, within some type of cell. If it's a bacteriophage, it needs to be within a bacteria cell. If it is a human-based virus, then it needs to be within a human cell, and on and on. So they require a living host cell. Remember, they're an obligate intracellular organism. So we are unable to just kind of swipe them onto an agar plate and then grow them. So infected host cells, whether or not they're eukaryotic or prokaryotic, can be cultured and grown, and then the growth medium can be harvested as a source of the virus. So what we do is we take different bacteria for bacteriophages and we grow those bacteria and we can infect them with a virus, and then we can then get the virus from that. Same thing with other eukaryotic cells. For example, human cells, we can grow them, infect them with a virus, and then we can get the virus out of those cells. So virions in the liquid medium can be separated from the host cells by either centrifugation or filtration. So centrifugation would be spinning it within a centrifuge, and then by spinning it very fast in a centrifuge, then it can separate out the parts. Or filtration, so there would need to be a small enough filter that would filter out things that are even slightly larger, like bacteria cells, which are actually very small, but definitely larger than viruses, from the viruses, and then just get the virus at the end. So filters can physically remove anything in the solution that's larger than the virions, and then the viruses can be collected in the filtrate. So there's actually an image of that right here. So basically, we have our solution up here, which can be, as they showed here, they used these red dots to be animal cells, the blue little spheres or circles to be bacteria cells, and then the green to be the viruses. So we've got this mixture of animal cells, of bacteria cells, and then of viruses. And if we have a filter pore size of 5 micrometers, then we filter out the red, which would be the animal cells, which are much larger than, say, bacteria and viruses. And then what we get in the filtrate, which is this down here, the solution that comes through, are bacteria cells and then viruses. But if we decrease that pore size to 0.2 micrometers, then we're going to filter out the blue and the red, which are the animal and the bacteria. And then what we end up with are just the viruses inside of the filtrate, which is the solution that has been filtered. All right, so then when we are cultivating viruses, we can do that in 2 ways. We can either do it in vivo, which is within a whole living organism, plant, or animal, so in vivo, or in vitro. So in vitro is outside of a living organism, so in an artificial environment like a test tube, a cell culture flask, or an agar plate. The way I remember this, which may be helpful to you, is in vitro has a T in it for test tube. That's how I remember it. Test tube in vitro versus in vivo is in the organism, a living whole organism. So bacteriophages can be grown in the presence of a dense layer of bacteria, which is called a bacterial lawn. So what we have is this what's called soft agar, which is 0.7% agar versus the 1.5% agar that is used in a typical agar plate. So we have this 0.7% soft agar in this petri dish, and then bacteria is grown on it. So for example, an E. coli that is not harmful, just a typical E. coli, can be grown in this soft agar in the petri dish, and then they can apply the bacteriophage to it. So this bacterial lawn would then be the E. coli over the entire thing. We wouldn't be isolating for individual colonies. It would just be a whole lawn, which is meaning completely covered. This can be grown in either a petri dish or what's called a flat flask or a horizontal flask. And we can see an image of that down here. So here's our typical petri dish or agar plate. So you can see kind of this creaminess here is the agar. You can see the creaminess in all 3 of these plates. And then this whole top layer is covered in bacteria. So we have a bacterial lawn on here. And then we'll get to what the dots are in a moment. But over here in A, these are the flat flasks. So they kind of stack on each other, which is handy. And then you can see the solution is right in here. And then that also allows for bacteria growth across the surface, but in just a little bit of a different way. All right, so the agar concentration is decreased, so it's called a soft agar. And since it is a soft agar, that allows the bacteriophages to diffuse through that medium. So remember, the bacteria or the bacteriophages need to burst out of the cell and then get to another cell. So if we had this 1.5% agar, then it's going to be too thick. It can't get through it. It's more solid. If we have a soft agar, then once the virions have been formed within the cell, they can burst out to become the mature viruses and then move on to another cell by getting through this soft agar. So for lytic bacteriophages, lysing of the bacterial hosts can then be observed in a clear zone called a plaque. And that's what we were seeing in those agar plates just a moment ago. So we had a plaque. And then this is as the phage kills the bacteria, then we see many plaques observed over this cloudy bacterial lawn. And that's exactly what we saw here. So we see this cloudy background, which is a mixture of the agar that's underneath, and then covering the whole surface, we have a bacterial lawn. And then all of these little dots that you can see here are called plaques. And that's where we have the bacteriophage that has lysed cells there. So there are no longer living cells or living colonies. So we've got these little empty spaces, and those are called plaques. So animal viruses require cells within a host animal or tissue culture cells that are derived from an animal, right? So we were just talking about bacteriophages. So we can grow bacteria. We know we can do that very simply and very easily in the laboratory, and they grow very well on agar plates, for example. Animal viruses are a little bit more difficult to deal with because we have to use actual animal tissue to grow them in. So animal virus cultivation is important for a couple of reasons. One, identification and diagnosis of pathogenic viruses in clinical specimens. So taking some cells and then filtering out the viruses, getting this virus filtrate, and then applying it to cells would be important to identify what kind of virus it is, also to diagnose in the clinic. Two, production of vaccines. So in order to produce vaccines, we need to produce a lot of the virus, and then we utilize that. We're going to talk a lot more about viruses or about vaccines in a later portion, but producing lots and lots of viruses is imperative to making vaccines, because then those viruses are adjusted in different ways to make different types of vaccines. So we need to have a lot of them. And then for three, in basic research studies. So in understanding viruses and understanding specific viruses, we need to be able to make a lot of them. Just like we see with bacteria. In order to study bacteria, we need to keep plating and plating and plating and growing up bacteria and utilizing them in the laboratory for research. So in vivo host sources can be a developing embryo and an embryonated bird's egg. So this is what's often used for an in vivo. Remember, in vivo is in an actual living organism. So for example, most of the flu vaccine, the influenza vaccines that are manufactured for the annual flu shot, annual flu vaccination programs, are cultured in hen's eggs. So they use an egg of a hen, and then they use it as an incubator to make these viruses, because remember, they have to be within a cell. So the embryo or the host animal serves as an incubator for this replication of the virus. So it can move from cell to cell to cell while it's incubating in the embryo. So location within the embryo or the host animal is important. So remember, we have this tissue tropism. So remember, what that means is that certain viruses will only go in and make copies of themselves and infect certain cells, certain tissue cells. So remember we spoke about brain cells specifically, so there's a virus that will only go into brain and nervous system cells, or there's a virus that will only go into liver cells. So many viruses have this tissue tropism, so it's very important that they be introduced into a specific site within this egg. So within the embryo, target sites include the amniotic cavity. So you can see here in our illustration, the amniotic cavity is right around the organism. So we have the embryo here. That's this guy here. So for example, in the hen's egg, it would be the growing chicken. And then we have the amniotic cavity right around there. So they made, like this one is injecting some filtrate with the virus in that amniotic cavity. Also, the chorioallantoic membrane. So the chorioallantoic membrane would be this region here, this membrane edge right there, which is another sac within the hen's egg. And then they can inject the filtrate, inject some virus right there on that membrane, and it can grow in the membrane. Or in the yolk sac. So the yolk sac is labeled here. It's this kind of yellow part. You are probably familiar with that in eggs, having the yolk. So it's the yellow portion there. So the viral infection, when they inject it, of course, may damage tissue membranes. So when they inject it, it may cause membranes, including lesions. A lesion due to a virus is called a pox. May disrupt embryonic development, so the embryo may no longer develop, or cause death of the embryo. So in this process, then the embryo likely will either not fully function or form correctly, or be killed in the process, either due to these pox, these lesions, or just because it's infected with the virus. So that was in vivo, right? So we were talking about in vivo, how to study animal viruses in vivo, and that would be using eggs and using it to cultivate. And we do see that used a lot in vaccinations. Specifically, every single year we have this flu vaccine that comes out, and that's done in hen's eggs. Now we'll talk a moment about in vitro studies. So we can study viruses in vitro. Remember, in vitro would be test tube. So that's outside of an actual living animal or plant. And we can use various types of cells for this to grow the viruses. Remember, there is tissue tropism, so having specific cells is important. Primary cell culture is then going to be freshly prepared from animal organs or tissues. So they're going to take those organs or take the tissues, and then they'll be able to prepare a cell culture from that. So the cells are extracted from the tissues either by mechanical scraping, which is basically exactly what it sounds like, scraping it off of the tissue and just gathering up some of those cells, or mincing to release cells. So either scraping off the cells and getting pieces of them, or mincing by basically putting them in a blender and breaking them up, and not too much because you don't want to lyse the cells themselves, but you want to kind of break up the tissue enough to get some cells free. Or by enzymatic methods, so using either trypsin or collagenase to break up the tissue and then release these individual cells into a suspension. So trypsin and collagenase are enzymes that are going to break apart tissues. Because of Anchorage dependence requirements, primary cell cultures require a liquid culture medium in a petri dish or tissue culture flask, so that they have a solid surface such as glass or plastic for attachment and growth. So Anchorage dependence is what it sounds like. It depends on something to anchor it. So these cells need to be able to touch something to anchor them down so that they can then go through mitosis. So primary cell cultures require a liquid culture medium, so some sort of solution to be in a petri dish, or this tissue culture flask. And then they need to have some sort of surface, like a glass or a plastic surface, to attach to and grow. So oftentimes there's a little bit of culture, a little bit of medium in there, and then it can still, actually the cells will be able to adhere to or attach to the surface of the petri dish or the flask. So primary cultures usually have a limited lifespan, so they are not able to live for very long. When these cells in the primary culture undergo mitosis, and then a sufficient density of cells is produced, so cells are produced going through mitosis, and they will spread and spread and spread, as each cell is undergoing mitosis, making daughter cells over and over and over again, then they'll come in contact with the next cell over that's been going through mitosis. When they come in contact with other cells, which is called cell-to-cell contact, then mitosis is triggered to stop. So we can put a certain number of cells in there. The cells will start to go through mitosis, but as soon as they touch another cell, then they stop, because in the bee, they wouldn't want to continue growing over another cell. They would just fill in that tissue area and then stop. So that's one of the cell mechanisms that's built into a cell, is that once it touches or abuts another cell, it stops growing. So this is called contact inhibition. So it inhibits growth when it has contact with another cell. So it prevents the density of cells from becoming too high. And we can actually see this down here in this illustration. So they have lung tissue, for example, and then individual cells have been isolated from the tissue, either through scraping them off of the lung tissue, trying to get a couple cells in this primary cell culture, or through doing a blender type of situation, or taking the lung tissue and adding those enzymes and getting some of the cells in here. So then we have our primary cell culture, which is a couple of cells, or several cells that they've isolated from the tissue, and then they'll start to undergo mitosis. So they adhere to the glass or the plastic here. They've got some solution with nutrients in it. And then we get to a point where they've gone through mitosis so many times that they now are at a point where they have contact inhibition. And so then they grow to the edge of the plate, and then they stop. And when they stop, then they're in a process of decay or a process of dying, so they're no longer useful. So to prevent this, to prevent contact inhibition, then the cells from the primary cell culture are going to be transferred to another vessel with some fresh growth medium. So here you can see they take some of the cells, they transfer them to a medium, and then we have our secondary cell culture at that point. And then those will do just like the primary cell culture does. They'll start going through mitosis, and then they will get to a point where they have contact inhibition as well, where they have stopped growing or stopped undergoing mitosis, and then we'll fill up this whole petri dish, and then they'll have to transfer new cells if they can. The thing is that cells, they can only divide so many times. So many cells, if they just, for example, this lung tissue, can only divide so many times before the cell will die. And then we have, cells in our body that are the same way. They undergo mitosis many times, and then they die, and then we have macrophages in our body that gobble them up, and we have plenty of cells behind that are filling in newer, fresher cells that are going through mitosis. So, I guess we'll talk about that in just a minute when we talk about Hela cells. But to finish up this thought here, we have our secondary cell culture. And then periodically, the cell density must be reduced by pouring off some cells and then adding fresh medium to provide space and nutrients to maintain cell growth. So that's what they'll do in the secondary culture. They'll try to pour some off, and then they'll add more medium in order to continue growing. So they'll try to get rid of some cells and then add more medium to grow the cells again. But again, once they reach the edges, then they'll stop going through mitosis and then be dying. So in contrast to primary cell cultures, continuous cell lines, usually derived from transformed cells or tumors, are often able to be subcultured many times or even grown indefinitely, which are called immortal cells. Continuous cell lines may not exhibit anchorage dependency. They can just grow in suspension, so they don't have to be in this single layer where they're able to touch the bottom and be anchored. And they may have lost their contact inhibition. So this continual cell line, so this is different here. Let me be clear. So for this example, we have our lung tissue. We've scraped some cells off, let's say. We've started growing in our primary cell culture, and then they grow and grow. Grow meaning go through mitosis. So they go through mitosis over and over again until we reach a point where they have cell-to-cell contact, and then they've stopped going through mitosis. We take some of these cells out, put them in a new petri dish, and then we have our secondary cell culture, which they can pour some out, add more medium, and keep them growing for a little while. Like I was saying, though, however, they will eventually die unless they are a continuous cell culture. A continuous cell culture, a continuous cell line, is usually from cells that have had some sort of change to make them be continuous or make them be immortal. And this is what we see down here. So let's say we have some lung tissue that has a tumor in it. So this is one of these changes, this transformed cell or a tumor. And we get these cells from the tumor, and we put them in our petri dish. And in this case, again, we don't see this anchorage issue. They can start to pile up and grow on top of each other. They don't have to be anchored down, and they don't have contact inhibition. So these can be extremely useful because they don't have to be babysat as much. They can grow over and over and over again. They go through mitosis over and over and over again, and they don't get to a point where they die. So they can grow in piles or lumps, resembling small tumor growths, which is why tumors are used, because tumors grow and grow and grow and grow. They don't have this contact inhibition, right? That's part of the problem with tumors, is that they don't stop growing once they bump into other cells. So these continuous cell lines are extremely useful in studying things like viruses, because then we don't have to keep, like I said, babysitting these other types of cells, and we can really do a lot of research because they just keep growing and they're immortal. So an example of this, I think the best example, the one that's most well known and has been used for many, many years, is the Hela cell line. And you should be familiar with this, which was originally cultivated from tumor cells obtained from Henrietta Lacks. So she had cervical cancer and died in 1951. She had cervical cancer, and she went to Johns Hopkins and tried to get some help from the doctors there. Well, they were doing research at the time, and so unbeknownst to her, actually, they took some of her cancer cells, and then they were trying to cultivate them because they were trying to do research on human cells, and they were not able to keep human cells alive. What they noticed then were the cells from her cancer, from her tumor, continued to live. They actually have an enzyme in them that keeps them from dying. It keeps repairing the DNA over and over again, and they've been immortal. So they've been in use and still are in use in thousands of places across the country and across the world since 1951. So these Hela cells were the first continuous tissue culture cell line. They were used to establish tissue culture as an important technology for research in cell biology, virology, and in medicine. So it's used across disciplines. So prior to the discovery of Hela cells, scientists were not able to establish tissue cultures with any reliability or stability. See, they would get to this point where they had cell-to-cell contact, and then they would start dying, and then the research would be gone, because then they were not able to get any viable viruses to study, because then without any functional living human cells, then the viruses would die because they have to be within the cells. So more than 6 decades later, this cell line is still alive and being used for medical research. All right.